Have you ever wondered how hospitals make sure that all of their surgical equipment are free from microorganisms? Have you wondered that? Okay. So, hi everyone, my name is Jess, and welcome to my YouTube channel, Med Ed with Jess, where we make medical lab science simple and easy to understand. So, into this lesson, we are going to be talking about types of sterilization. We'll talk about sterilization, the definition, the types, and the application. So stick around. Let's get into the video. Start with the definition of sterilization. Sterilization is actually a process where we remove or we kill microorganisms from objects or instruments or anything that we are using in the lab, in the hospital. Yeah. So the process in which we make those things free from microorganisms is known as sterilization. Sterilization. Sterilization is the complete removal of microorganisms such as bacteria, um, viruses, or even fungi and bacterial spores. So it is the complete removal of microorganisms from a surface or an object. Yeah. Most students actually confuse sterilization with disinfection. So let me tell you the difference between sterilization and disinfection. So we have sterilization here, sterilization, and disinfection here. When we are talking about sterilization, we are actually talking about the process where we completely remove all of the microorganisms, all of the microorganisms, all of the microbes are removed from on that object or from on that instrument we are trying to sterilize. So sterilization means complete removal of microorganisms. But when we are talking about disinfection, disinfection is the removal of the harmful organisms, the harmful ones, right? And disinfection is mostly done, on non-living objects, right? Like for example, right, flow on the floor. We use disinfectant for flow, for chair, and etc. in the laboratory. So disinfection and sterilization are two different things. Not confuse them. And sterilization is mostly used for instruments, for instruments for the treatment of biomedical waste. That's where we use sterilization. For disinfection, it's used for floor, for chair, for benches, in the hospital setting. So now let's get into business. Let's talk about the types of sterilization. There are basically four main types of sterilization that we are going to be discussing or covering into this lesson. So let's get into them one after another. Firstly, we have dry heat sterilization. The first type of sterilization is known as dry heat sterilization. So dry heat sterilization is the first type of sterilization. And now you might be thinking, what do we do in dry heat? So dry heat sterilization actually kill microorganisms using something we call oxidation. When I'm talking about the difference between disinfection, in another video we explain details about oxidation. So make sure to subscribe and come back for another video, right? So dry heat sterilization, it's a process where microorganisms are killed using oxidation. Example of dry heat sterilization include hot air oven. So hot air oven is an example of dry heat sterilization. And then we also have flaming. And then we have another type of dry heat sterilization is incineration. Incineration. Right. So these are the types of sterilization that we have when it comes to dry heat. Example of it, we have hot air oven, we have flaming incineration. So now for hot air oven is an instrument that is used in the laboratory is an instrument that is used in the laboratory to be able to get rid of microorganisms on surgical instruments, in cultural media, and etc. And then we also have flaming, like you can use the flame to get rid of microorganisms and incineration by burning things also. So dry heat sterilization actually based on the principle of oxidation. So now let's talk about the application of dry heat sterilization. Application of dry heat sterilization. The application. Now, application application of dry heat. It can be used to sterilize glassware. Do you know what we mean by glassware? Glassware are equipment that we use in the laboratory, like your beaker, your cylinder, all of those things that you use to perform your laboratory work. They are called glassware, made of glass, right? And then we can also use metals. The application of this, you use oil, powder, sterilizing powder. If you want to sterilize powder, glassware, metal, oil in the laboratory, we go for dry heat sterilization method, which include your hot air oven, which also include flaming or incineration, right? So these are all part of dry heat sterilization method. Now let's move to the next one. The next one is called moist heat sterilization. Now we are done with the dry, so we move to the moist. We have the moist, MOIST, the moist heat sterilization. So now in this moist heat works on the principle of denaturing the protein. It kill microorganism by denaturing the protein that is present inside of the microorganism. It will denature the protein. And one of the most common example of moist heat sterilization is autoclave. Autoclave. We have autoclave. Autoclave is actually an instrument that is used in the laboratory to carry on sterilization, and then we use it at 140 degrees Celsius for 15 minutes, 15 to 20 minutes at a pressure of 15 pressure. So this is an example of moist heat sterilization. So now let's talk about the application, right? Moist heat sterilization, remember, it based on the principle of denaturing the microorganism, denaturing the protein present inside of the microorganisms. And then we have an example of it is autoclave. An autoclave is an instrument that is used in the laboratory to be able to carry on sterilization. It is used at 121 degrees Celsius for 15 minutes at 15 pressure. Remember that because it's very important for your examination. So moving on, let's talk about the application of moist heat sterilization. Application of moist heat sterilization. So it can be used to sterilize cultural media, surgical instrument, these are the few applications of moist heat sterilization, which is actually autoclave. So so now we have it can be used to sterilize cultural media. It can also be used to sterilize surgical instruments and laboratory equipment also. You can also add another one here. It can be used for laboratory equipment. Um, It can be used. It can be used also to sterilize laboratory equipment. So these are all of the application of moist heat sterilization. So remember, we have talked about two now, right? Let's move on to the next type of sterilization. The next type of sterilization is a very important or common one that uh it's used mostly in the hospital. We have radiation. We have the radiation sterilization. Radiation, radiation sterilization, sterilization. So now, this is the process of using radiation to sterilize instruments in the hospital, right? Or materials. So there are two types. We have the UV, you know, the UV ray, and we have the gamma. In another video, I will explain both of them in detail, but today it's just an overview of it, right? So we have the UV ray and also the gamma ray, right? So the UV ray is actually used to disinfect. It is used to disinfect laboratory, hospital benches, chairs, and etc. And it have a very poor penetrating power. It have a poor penetrating power, so it does not really kill all of the microorganisms. It just kill the harmful ones, right? And then now, gamma ray, gamma ray is actually used in biomedical ways, right? Like if you want to get it, it is used in biomedical waste treatment. Biomedical waste treatment, we use the gamma ray. If we want to treat biomedical waste, it is used for the treatment of catheters, surgical instruments, syringes, and all of those things. We use the gamma ray. So let's move on to the last type of surgical. Let's move on to the last type of sterilization. So remember, radiation sterilization has two types. We have the UV ray and the gamma ray. UV ray is just used for disinfection. It have low penetrating power. Why the gamma ray have or strong or high penetrating power, and it's used in the treatment of biomedical waste. So the last type of sterilization is actually called filtration. Filtration sterilization. Filtration sterilization is the type of sterilization where we just filter the liquid. We actually filter the liquid to get rid of the microorganisms. Because if we are actually using heat sensitive material or heat sensitive liquid, then we go for filtration sterilization. Like for example, right, it can be used in vaccines. We can see it in vaccines. It can be used in antibiotics solution, and also some heat sensitive, heat sensitive liquid. So this is the application, right? So these are all of the types of sterilization. So let's do a recap about the types of sterilization before we can talk about the factor that affects sterilization and the application of sterilization. Now, the first type is dry, dry heat. The next one is moist heat. The next one is radiation. Lastly, we have filtration. So these are the types of sterilization. Dry heat, an example of dry heat is hot air oven. We have hot air oven. Another one for moist heat, we have autoclave. For radiation, we have two types. We have the gamma ray and the UV ray. Lastly, for filtration, filtration is actually used for heat sensitive liquid. Do not forget. Yeah. So this is it. We have dry heat, moist heat, radiation, filtration, and that's all. These are the types of sterilization that is used in microbiology lab, using the hospital lab to be able to sterilize equipment, cultural media, surgical tools, and everything. So now let's talk about the factors that affect sterilization. Factors affecting sterilization. Now I'm going to be telling you some that affect that affect. This is C, guys. Sterilization. Factors that affect sterilization. One of the first factors that actually affects sterilization is the temperature. The temperature at which sterilization is performed actually affects sterilization. Remember the temperature. We also have the load of microorganisms that you are trying to get rid of, right? The load of microbes also affect sterilization. And then the method you are using. One of the most effective methods is actually moist the moist heat which you use to autoclave. Yes, the sterilization is the time taken to sterilize that material, right? Remember, for example, autoclave we have 15 to 20 minutes, and the nature of bacteria that you are trying to get rid of, the microorganism involved, right? So these are the factors that affect sterilization. So now, lastly, let's talk about the actually application, right? The application of sterilization as the last part of today's lesson. Let's conclude with the types of the application of sterilization. Application. Application. Sterilization is used in hospitals, in laboratories. It is also using food industries and even in the pharmaceuticals industry too. Sorry guys. So it is used in the hospital, it is used in the laboratory, it is used in food industry, it is used in the pharmaceutical industry too, to be able to get rid of microorganisms in um get rid of microorganisms where they are present. Right? So let's talk about a few exam tips, right? Examination tips. First of all, remember this, right? There are basically four types of sterilization. We have heat, moist, we have dry heat, we have dry heat, we have moist heat, we have radiation, we also have filtration. Dry heat works on the principle of oxidation, which include hot air oven, flaming, incineration, etc. Secondly, we also have moist heat, which actually work on the principle of denaturing the protein of the microorganism. An example is autoclave, right? Remember that autoclave work at 121 degrees Celsius for 15 to 20 minutes at around 15 pressure. Then we have filtration, which is used for heat sensitive liquid in the lab. And also, we also have the last one, which is radiation. There are two types of radiation that is used. We have the UV ray, which is basically used to disinfect benches, chairs, like that, right? And you have pore penetrating power. Then we have the gamma ray, which is also used to disinfect or to get rid of microorganisms that are found in the catheter, the syringes, all of those things. Biomedical waste treatment we use gamma ray also. So we have a strong or hard penetrating power. So this is all of the lesson. So so this is everything that we have for today. We have come to the end of today's lesson. Um, if you enjoy the lesson, do well to like the video, subscribe, and don't forget to share with your classmates so that they can understand the types of sterilization too, right? Until then, I'll see you on the next video. So keep learning, keep studying, and keep growing. Bye.